Hi, I'm Dr. Kavina Ganapathy from Department of Biotechnology, School of Sciences, JAIN deemed to be university. As a researcher working at the intersection of cancer biology and therapeutic innovation, I often get asked by people why cancer cells are very difficult to be treated. Well, we all have heard the word cancer, but what lies behind is far more complex biological story of cells, which has an uncontrollable division and invade immune system as well as migrate to places where they don't belong, which results in the imbalance in the homeostasis and also bring in difficulty in therapeutic treatments with the resistance of treatment strategies. What makes cancer difficult to treat is not how fast it grows, but how smart it is to adapt, learn to resist treatments, and also to reprogram the immune defense so they can hide for a longer time inside the body, escaping the treatments and as well as spread, which is called metastasis. What is far more interesting is the recent light on cancer studies in space. Well, I'm talking about Axom Mission. Well, people might wonder what it has to do with cancer in space research. I would like to walk you through this cancer research in the space research especially. In Axom Mission 1, what they did is they have taken the human cancer cell lines in the nano vessel with the medium and they have assessed the growth of cancer cells. Well, interestingly, what they found out is that these cancer cells are robust growing in microgravity, and also they activate the survival genes, something like ADAR1, which aids in the robust growth of cancers, which was not very evident in the gravitational studies, which is very interesting. And in the second Axom mission and also the third, they assess the efficacy of two drugs. I will not take the names of the drugs here, but I will explain what the drug 1 is. Drug 1 is an investigational drug and the other drug 2 is an FDA approved drug. These two drugs were assessed on the robust growth of cancer. And again, the interesting fact about the studies carrying with the drugs are the investigational drug 1, which was a derivative of a natural product, that is from a bacteria, and which was found to inhibit the ADAR1 gene. I told you ADAR1 gene is the one which was discovered in the Axom Mission 1, which aids in the robust growth of cancer cells. By inhibiting this ADAR1 gene, the growth of the cancer cells was inhibited by this drug 1 compared to the other drug. This innovation was very interesting to know because it is slightly different from the gravitational study, which shows the drug enhancement or efficacy in the microgravity, which brings in a combinatorial study or an adjunct treatment strategies to cure cancer. Maybe. So what is interesting about this space research is this Axom Mission 4, which has 60 scientific experiments planned with the activities of 31 countries. And one among the experiments is again the cancer cells. And this time they have carried the organoids from triple negative breast cancer cell lines. When I say triple negative, that means it is estrogen receptor negative, progesterone receptor negative, and the human epidermal growth factor receptor negative. So this makes the cancer organoid very resistant to treatments and makes it a stubborn cancer organoid maybe. And they have assessed these two drugs again on these organoids. They are planning to give us the data of what exactly happens to the drug resistant cells with respect to the organoid model. And also they are assessing the early diagnostic markers if they can find something in the microgravity. Researchers like me are very inspired because we do all regular lab assessments on the cancer activity. In my lab, what we work on is the organoid model, where we assess the tumor microenvironment with respect to extracellular matrix, stromal cells, immune cells, as well as endothelial cells mixed to form something very similar to the tumor what you find in the in vivo models. And we also assess the therapeutic components, the bioactive components from plants and different sources to figure out their anti-cancer activity with respect to the cancer cell lines what is available or the model system. And we also work on the patient organ systems, organoid tumor model systems. Having said that, one can ask me how exactly is this bioactive components are assessed to see that there is a progress in the cancer treatment or not, or what kind of experiment is designed to show there is a potential candidate for therapy. So what we do is a multi-layer assessment on different cellular and molecular levels of cancer by after treatment with the bioactive components. So once the treatment is done, we assess the DNA damage. We also assess mitochondrial function. We assess apoptotic and migrational pathways. We do transcriptomic analysis using RNA sequencing. Overall, all these experiments show us whether our drug is capable to induce tumor death or a potential candidate for treatment of or innovation in the cancer field to design a personalized cancer treatments for different kinds of cancers. One of the most insightful tools, that is the Comet Assay, which we regularly use in the lab, is to assess several bioactive components to see whether they actually have the ability to kill cancer, and especially to assess the DNA damage. So the Comet Assay is affordable, it's not time consuming, and we can work on a limited resources which we have it in lab to find out a novel result of whether the candidate is eligible for the treatment or not. A Comet Assay is first described by Olson and Johnson in 1984, and this assay gives you a single cell analysis of DNA damage, which is also called a single cell gel electrophoresis, where you can assess the DNA fragmentation at the single cell level. What we do is we embed the cells in the gel and run it through electrophoresis and stain and see whether they show the DNA damage. How do we see the DNA damage? Because it forms a tail which is similar to the comet what you see. That's the reason why it is called Comet Assay. We can give you a quick demonstration of how it is done in the lab to figure out how insightful the tool is. This is Varsha Vinay Kumar. She is the research intern in Cancer and Regenerative Lab, and she'll be assisting in doing Comet Assay. This is the slide we have, which is square in shape for Comet Assay, and we have cover glasses which has to be wiped clean with 70% ethanol. Once the slide is clean, we have to prepare 1% agarose gel. And for 1% agarose gel, we have to weigh 1 gram of agarose in 100 ml of TE buffer. TE buffer is a tris EDTA buffer with pH 8.3. Once the agarose is added, we have to heat it in the microwave at 80 degrees Celsius for 1 minute. After it is melted, you're supposed to pour it on the clean glass slide evenly and uniformly. So once it is added uniformly, we have to place the cover slip on the gel and let it solidify. So meanwhile, we prepare the cell suspension for embedding the cells inside the agarose. So we do have two culture dishes. One is control and one is treatment. Control do not have any drug treatment. And the treatment culture flask has a drug bioactive component, which may be possibly the candidate for cancer therapy. And to assess the cell DNA damage, we have to take a cell suspension, which the cell density for the use is 2 lakh per assay. So 2 lakh cells are counted using the cell counter, which is already ready and kept ready for mixing it with a low melting agar. So this 2 lakh cells will be mixed with the low melting agarose, and this is a purified and modified version of the agarose where it has a lower melting point and also the setting point is also low, which does not damage the cells. We have to be really quick because the setting point is 37 degrees Celsius. So once the lower melting agarose is mixed with the cell suspension, we layer it on the 1% agarose plate which is already coated, and we place the clean cover slip on the assay. We let it settle for 20 minutes in 4 degrees Celsius. Now the gel is set. Once the gel is set, we have to carefully remove the cover slips. After the cover slip is removed, we are supposed to add the lysis buffer. This lysis buffer contains a detergent called Triton X, which creates a pore in the cell membrane, which perforates the cell membrane of nucleus as well as the cell as such, which lets the content of the nucleus to flow through it when the gel electrophoresis. If there is the DNA damage, the fragments of the DNA flows through, giving you a comet-like appearance. And after the lysis buffer is added, we are keeping it for 1 hour incubation at room temperature, 37 degrees Celsius. Once the lysis is done, after an hour of incubation, the lysis buffer is discarded and we add alkaline buffer. An alkaline buffer is added because it unwinds the DNA. It breaks the hydrogen bonds between the base pairs, makes the double-strand DNA a single strand, and it is easier to assess the fragments which moves out of the perforated membrane. Alkaline buffer is added for 20 minutes at room temperature. After the incubation at 20 minutes, we run with the same alkaline buffer in electrophoresis at 25 volts for 30 minutes. After the 30 minutes of electrophoresis, we have to put neutralization buffer. Neutralization buffer is very important because it stops the denaturation of the DNA. And once it is just the washes for 3 minutes. Once the neutralization buffer for 3 minutes incubation is done, we are adding 5% Giemsa stain to stain the nucleic proteins and chromatins of the DNA or DNA fragments. % Giemsa stain is added onto the slice and incubated for 15 minutes in the room temperature. Once the staining is done, the gel is washed and then taken under the trinocular microscope to image the comet. Let us now visualize the treated cells for the DNA fragmentation. After the staining with Giemsa, you can see the DNA damage is depicted by the tail-like structure here, which is comet-like. You can see the comet-like shapes from each cell, which shows the damage of the DNA fragmentation. The longer the tail shows the intensity of the DNA damage. And now we will visualize the control. As you can see, there is an absence of the tail, comet-like structures in the control, showing that the bioactive components has worked for the DNA fragmentation, and this is how we do initial assessments using a Comet assay. I would like to conclude by saying that the fusion of Earth-bound research and the space research has opened new doors for two-way learning to outsmart cancer before it outsmarts us. Let's build bridges across disciplines, distances, and even gravity. Thank you for listening.